Now, let's talk about something about gene therapy. Human diseases can be driven by the alteration of one or more genes, including via gene amplification, mutation, and gene fusion, et al. Gene therapy promotes the production of desirable or therapeutic proteins and holds great promise. The first gene therapy is human myoblast genome therapy for hereditary degenerative diseases, which is conducted in 1990. Since then, many efforts have been made in the gene therapy area. In 2003, the Chinese State Food and Drug Administration approved Gendicine, the first cancer gene therapy product which is indicated for head and neck cancer therapy. Although it goes through numerous ups and downs, gene therapy has come of age. So far, more than 40 gene therapy products have been approved globally. TP53 is a famous tumor suppressor gene entitled the guardian of the genome. A variety of putative p53-mediated tumor suppression mechanisms have been described, such as inhibiting cell growth, reducing metastasis, regulating metabolic switching, remodeling the cancer immune microenvironment, and inducing cancer cell apoptosis, autophagy, ferroptosis, and cellular senescence. Genetic mutations in TP53 contribute to human malignancies through various means. TP53 gene mutations stimulate hematopoiesis and tumor metastasis, promoting a more aggressive phenotype for the tumor. Moreover, TP53 mutations have an impact on both the tumor's response to treatment and its survival following exposure to a variety of stresses. To date, there have been a variety of therapeutic strategies targeting p53, including gene therapy to restore normal p53 function, mutant p53 rescue, inhibiting the MDM2-p53 interaction, p53-based vaccines, and several other approaches. This review focuses on the functions of TP53 and discusses the aberrant roles of mutant p53 in various types of cancer. Gendicine has made tremendous progress in the field of cancer gene therapy. The recombinant human p53 adenovirus injection was developed by Sibiono Genentech Co. and first described publicly in March 1998. There have now been at least 52 clinical trial protocols utilizing recombinant human p53 adenovirus products worldwide for the treatment of 26 different malignant tumors. Based on 20 years of commercial use in more than 30,000 patients and more than 30 published clinical studies, Gendicine has demonstrated an impeccable safety profile. We herein describe the clinical responses reported in clinical trials and summarize the methods of Gendicine administration for different cancer types to provide a reference for future clinical applications. Looking back at the history of gene therapy development, we have entered a new era, as evidenced by an expanded spectrum of clinical indications, more sophisticated approaches for gene editing, as well as more optimized vectors. Our team focuses on precision cancer therapy and the rationality of the combined application of clinical drugs. We have established a mature system for cancer precision therapy, from molecular mechanism research, animal models, human organoid library, and bioinformatic analysis, to clinical trials. We bring together cell biologists, clinicians, and bioengineers to achieve our goals.